Students, in this video lecture, we will talk about the amplification of gene by PCR, polymerase chain reaction, and large scale protein production, means large scale recombinant protein production, and downstream processing of the recombinant protein. So first we will talk about the amplification of gene of interest by PCR. PCR means the polymerase chain reaction. In this reaction, the multiple copies of a gene, or we can say the fragment of DNA of interest of desired size and the desired location, is synthesized in vitro using some components. It means if we want to get a small piece of DNA, so we can synthesize it synthetically in different size, and it may present at different location. Means at different location we can get a synthetic fragment of DNA by using this technique, which is known as PCR, polymerase chain reaction. So now we will see what are the components we need to use in this reaction. First one is primer. So primers are small chemically synthesized single-stranded DNA, and these primers are complementary to both the borders of the desired gene. Suppose I want to amplify a particular gene, suppose a growth hormone. So I need two primers. One will bind to the one end of the growth hormone, and another will on the another end of the that DNA fragment, means growth hormone. And generally these are present as a single stranded DNA, and the length of this DNA ranges from 18 to 30. Principally it should be 20, and as I told you, it should present in the pair. One border's complementary primer will be called as forward, and another primer will be called as reverse primer. And the second component is DNA polymerase. The DNA polymerase is an enzyme which extends the primer on the genomic DNA by adding nucleotides, and that nucleotide will make the complete strand of the DNA. Then the third is DNTP, dinucleotriphosphate, and as we know, there are a total of 4 nitrogenous bases in DNA. So there are 4 different DNTPs. One is DATP, DGTP, GCTP, and DTTP. Means all the bases will have different DNTPs, and all the 4 DNTPs will be mixed together. And the fourth is buffer. The buffer is the combination of some salts and chemicals which provide an environment for the extension of new strands. And the fifth is the genomic DNA, means the DNA from where we are going to amplify, or we are going to make the synthetic DNA strand. Suppose I am going to amplify the uh, or going to synthesize the growth hormone protein of uh, cattle, then I need to have the genomic DNA of cattle only. So now we will see what are the conditions we need to provide to get the desired fragment of DNA. So the conditions are, so above 5 we have seen 5 components: primer, DNTPs, DNA polymerase, buffer, and genomic DNA. They will be mixed in a tube, and they will be exposed to a different temperature conditions on a machine, and that machine is called as thermocycler. And the condition may be there is mainly uh, 4 conditions. One is initiation denaturation. It means we will provide a high temperature and it will be around 94 to 95 degrees centigrade for 3 minutes. And the second is, and this initiation denaturation we will provide of only one cycle, means we will provide 94 degrees centigrade for 3 minutes only to the all the components. Again, what will happen, we will provide denaturation, means 94 degrees centigrade for 30 seconds. Then just after that, we will provide 60 degrees centigrade for 30 seconds and 72 degrees centigrade for 1 minute per 1000 base pair. It means for the preparation of 1000 base pair of DNA, we should provide 1 minute. If I want to amplify or I want to get the desired DNA fragment of 2000 base pair, then I will provide 72 degrees centigrade for 2 minutes. And this this cycle will be repeated, means again 94 degrees centigrade, 60 degrees centigrade, 72 degrees centigrade. Again, the cycle will be repeated, and a total of 35 to 40 cycles we used to run. Means we will provide the different temperature in the thermocycler to all the uh, components. Now we will see what is the role of this temperature for these components. Now we can see, suppose this is the long stretch of genomic DNA, and this is the location of, suppose, growth hormone or insulin gene. Suppose I am going to amplify the insulin gene. So suppose this is the location of insulin gene and this is the border of this insulin gene. Now we will zoom it. So this is the border of insulin gene out of the total genomic DNA, and this is the one strand of genomic DNA. So what happens when we will provide denaturation, means 94 degrees centigrade, then the hydrogen bond between these both of the DNA strands will be broken. Now we can see both of the strands have been separated here. Now we will provide the another temperature, annealing, and generally it is around 60 degrees centigrade. When we will provide 60 degrees centigrade, then this condition helps the this oligonucleotide or primer to bind its complementary region. So the primers will scan its uh, complementary region in genomic DNA and they will get bind. So we can see here our forward primer has bind on this strand and the reverse here. We can see here one primer will bind to the one border of the genomic DNA and another will bind to the another strand of the genomic gene. We can see here. So at the annealing temperature, primers get bind. Now we will provide this uh, this system to 72 degrees centigrade. Then at 72 degrees centigrade, we can see here the DNA polymerase will bind to the both of the primers and it will start extension, means it will start moving in the direction of 5 degree to 3 degree, means 5 dash to 3 dash direction, and it will start adding nucleotides. This condition is called as extension just because the we can see here the primer is now extending itself on this genomic DNA strand. So it will start adding uh, new nucleotides as per the complementary region. So it will add different nucleotides here, and it will form. We can see here at the end of this 72 degrees centigrade. We can see here now we had started with one strand. Now we can see the result is two strand DNA, means at the end of one cycle two strand has been formed. In the same way, at the end of second cycle, four strands will be formed, and after the 40 cycles, billions of the copies of the genomic DNA will be formed. So by this, we can get the synthetic DNA strand synthetic DNA fragment of our desired DNA fragment, which can present at any region of the DNA. Now we will see how we can get the recombinant protein in large scale or industrial scale. So the ultimate aim of the insert of the desired gene into the vector is to collect protein from the bacteria or animal or plant or yeast cell, or we can say the host cell. We have already discussed how we used to insert our recombinant DNA into the host to get the desired protein, desired recombinant protein. The protein expressed from the gene of the another animal, plant or organism source is called recombinant protein. For the large scale recombinant protein production, specific techniques are tools and techniques are needed. And in large scale protein production, we used to use bioreactors in which we grow a large volume of host cells with recombinant DNA, and it is around 100 to 1000 liters of the amount. It means we used to grow 100 to 1000 liters of the host cells with our recombinant DNA, and from there we used to collect our recombinant protein. The bioreactors are nothing but just a large vessel in which large raw materials are biologically converted into specific product or enzymes, etc. So bioreactors provide an optimum condition for the achievement of desired products at definite pH, different temperature, and salt and oxygen, etc. It means altogether it will provide a specific optimum condition to produce a desired amount of recombinant protein. So now we can see there are mainly two types of bioreactors. One is stirred tank and another is sparged stir tank. So we can see here there are two propellers are, or we can say the flat blades for the steering. Means we used to put the cells here, a host cell with the recombinant DNA, and this blades are moving in clock or anti-clockwise, so that a proper mixing of and proper mixing and aeration can be provided, so that a large amount of products we can get. And here in this case, the sparged steer tank, the same happens. Means we used to use a propeller here, and this propeller not only uh, rotates in clock or on anti-clockwise to mix the whole system, but also it also provides a spark of air also. This air helps for the for providing oxygen to the cultured cells, and in some cases, sparge steer tank is better as compared to steer tank, just because it also provides oxygen also. So the surface area also increases, and the production of recombinant protein will be promoted here. So to maintain the cells in the log phase, the used media is consistently removed, and the removed media is replaced by new nutrient media, so that the cells will be remain in the log phase. After the production of recombinant protein, we used to go through downstream processes. So after a growth of cells, a lot of step-by-step processes are also followed to collect the product in desired form up to reach in the market. So we will see what are the processes. First is the separation and purification of specific desired protein or recombinant protein. Means the host will also produce some non-essential proteins also. So we need to separate our recombinant protein out of the total protein. So it will be called as separation. Again, the purification means we need to get a pure form of recombinant protein. So we will utilize some technologies and different techniques to purify that recombinant protein. As we know that it will be used as a human in the human being. So it should be a very pure. And the second step is preservation at specific condition as to prevent any compromisation in their quality and activity. We need to preserve it at a specific condition, means at 4 degrees centigrade or minus 80 degrees centigrade, or if we know that this protein product will be remain intact or remain functional in this buffer, so we will provide a specific condition. So preservation is also an essential step. And the last is quality control testing. Means we need to test each and every time the quality of the recombinant protein so that we can utilize the recombinant protein for the human consumption. Thank you.